say a man. A man can take somebody's egg, and then this is all human. Am I making some sense? It's all human. You can take the egg and take the nucleus out, and this man take his liver cells, any cell in the body that has all 46 chromosomes, and take the nucleus out and put it in here, and they put it in a dish. Well, they give it a shock, and they put it in a dish, and it becomes a early embryo, a blastula, blastocyst. That's in case of human, and then they put that blastocyst in a liquid nitrogen. Am I making some sense, everybody? Later on, that person had too much partying, and he needs liver cells. Is that right? They take cells out of liquid nitrogen right here, and then they will go ahead from here. They take the cells and grow it and have liver cells right here, and they infuse it into that map. Does that make sense? So cloning and stem cell, it can go hand in hand based on what you have, your PowerPoint. What do you have? Am I making some sense to everybody? So we talked about all of that. And then we talked about Carlos Linnaeus and Robert Whittaker. Is that right? Is that really right? And then Carlos Linnaeus, he proposed that. And then we talked about human. Is that right? Classification. I don't think I asked morning lab. Did I ask any quiz question about this classification? Human classification. What is the name of the class? What is the name of the order for human? Okay, well, you're like, not you, I don't know. Maybe you studied it and I didn't ask. But it might appear, it will appear in your, I don't know, next quiz, exam, whatever. You should know this. And this is where we stopped this morning. Is that right? We stopped right here, and we were talking about Protista. They are a group by themselves. They own their own kingdom. And the first life appeared on Earth based on your textbook 3.5 billion years ago. There is a new research out that they're indicating it was 7 billion or something like that. Yeah, 7 billion. That's what I heard on the radio. 7 billion years ago. So I'm not going to hold you responsible for those numbers because they change. But that's pretty much life was the very first one. And the first one still, it's unanimous among scientists that the first life was a bacteria. It was an archaebacteria, or what do you call it? A prokaryote. Okay, a prokaryote. It does not have nucleus. Then kingdom Protista appeared roughly about 1 billion years ago, they say. And then Protozoa and subkingdom, you might see those terms interchangeably. Protista, Protozoan. They are animal-like because some of them do not have cell wall. In some ways, they are motile, some of them, and ingest their food. That's what they are animal-like. I didn't write down they are plant-like also, like volvox you're dealing with in the lab. Euglena, I will talk about it. They do photosynthesis. Okay, they do not engulf their food. Okay, so some act like plants and some act like animal. So what are they? That's why scientists start putting them in their own group. At the beginning, I want to say at the beginning, when during Carlos Linnaeus, how about that? Or 100 years ago, 150 years ago, there was just two groups of living organisms, plants and animals. Nowadays, we have a whole bunch of them. We have the archaea and eukaryote and archaebacteria and then archaebacteria and so on and so forth. Okay, we will discuss these phyla, the main characteristics of protozoa phyla. It's the unicellular, the microscopic, no germ layers. When I say germ layer, what comes to your mind? What should come to your mind? Very good. Ectoderm, endoderm, mesoderm. If somebody grab your collar in the mall and say germ layer, say ectoderm, endoderm, mesoderm, they will let your collar go. They will let your collar go.
And then I'll get a restraining order.
And then, yes, they won't get a restraining order. So germ layers, they do not have any germ layers. Specialized organelles, you will see them in a minute. Some species, they have some specialized organelles. One of them, I'm going to give you an example, it's on top of my head right now. It is apical complex. They call it apical complex. It's one of the specialized organelles in there. So they have free living, mutualism, and commensalism, and parasitism. Can you guys from Bio 1 refresh my memory what they were? I mean, I hope in the bottom you can see that. William, can this bottom show up here? Or should I hold the screen right here?
It's good, yeah.
It's good. Okay. So free living, they're free living. Parasite or the term symbiosis. The term symbiosis. It means what? Does anybody know? Living together. Thank you. So when you have a mother and a fetus, right? A mother and the baby in the womb. Is that an example of a good example of symbiosis? They're living together.
No. It's when they help each other.
Because the definition, you said a very short, very good definition. That's what you said. Sim together, bio it means life, living together. Is that right? Life, living together. But a good definition of symbiosis means two species that they live together, as you said, but they are different. They're different species. Okay, so if a woman is pregnant with a donkey, then that's symbiosis. You can call that a symbiosis. Okay, so there are different types of, there are four actually, but we'll emphasize on three of them. There are three different types of symbiosis. One of them is called mutualism, and what does that mean? Huh? It means they both benefit from this living together, like termite gut. In the termite gut, there are some dinoflagellates. Well, you're going to study them. Trichonympha here. That's what I was talking about. Trichonympha in the termite gut breaks down cellulose, the wood they eat, the termites eat, to glucose, both for the termite and for the trichonympha. And then, what is commensalism? Anybody? What does that mean? Huh? One benefits and the other one, I'm going to just draw a multiply sign. It's not hard. Okay? A good example of that I have for you, Entamoeba coli in your gut is not supposed to harm you. It's not supposed to benefit you. It's just there, but they're benefiting. The E. coli, Entamoeba coli, not the bacteria E. coli, Entamoeba coli, you will see it. It's not harming you, but the E. coli is benefiting. Okay, the other one is parasitism. Right? Parasitism, what happens? One benefits and the other one is harmed. Not necessarily die, yes, they could eventually die. Yes, you're right. But one is benefited and the other one is harmed. Now, if I have to ask you an evolution question right here, of these three, which one you think came first? Which one, after a few million years, came second, and which one came last? What do you think, and why?
Parasitism first.
Parasitism first. And what is your logic?
Take more time to be mutually beneficial, so it's kind of more akin to eating or ingesting.
Okay. And then what happened next? What is the next evolution? Commensalism. And why is that? And of course, law of elimination, is that right? Mutualism is last. Right? So yeah, let's say that's true. Why?
They have to evolve together.
Yeah, they evolved together, it's an armed race, and what happened? What is the logic? Anybody? What happened here in case of parasitism? One was benefiting and the other one would be harmed and eventually die, right? So if this host dies, if that symbiont, they call it. Symbiont, symbiont. This symbiont dies, then this one will die too, right? Just pure fact. So over millions of years, this probably was 500 million years ago, if I have to give it a number. 500 million years ago, that was the case. 250 million years later, they learned not to harm each other. The symbiont learned not to harm it, but just let it be. Let that poor symbiont be. And finally, they learned present time, if we have to, the present time they both benefit. So they can both live longer. It's just not this guy who lives. Now they both live. And I gave you one example of each. Am I making some sense? So that was pretty much the case between this evolution of symbiosis. Some sessiles, some Protista or Protozoa or sessiles, some they move by pseudopod, some move by flagella, some move by cilia, and then in Bio 1, you learn them all. In Bio 1, you learn them all. That's what each one of them they do. Okay, all types of nutrition, autotroph, heterotroph. Autotroph, it means what? They make their own food. And heterotroph means what? Yeah, we are heterotrophs. Is that right? You're different. You're different. We have other things to eat. We cannot make our own food. When you make your own food, that is autotroph, photosynthesis. Aquatic and terrestrial, all sort of organisms, asexual and sexual reproduction. You know, you have some species multiplied by both, and so on and so forth. They have most of the organelles that are other eukaryotic cells have mitochondria. Some do not have mitochondria. Endoplasmic reticulum, Golgi apparatus, vesicles, chloroplast, vacuoles. Again, not all of protozoa have all of these organelles. I hope I'm making some sense. You all know what I'm talking about. Not all of them have. But one thing they all have. What is that, you think?
Cell membrane.
Yeah, cell membrane. Bacteria have cell membrane too. But one thing they all have. Nucleus. They're all eukaryotic. They're all eukaryotic. They all have nucleus. Okay, ectoplasm, endoplasm, if they move by a pseudopod, locomotion, cilia, you learned that in biology, 1, 9 plus 2, is that right? 9 microtubules in the parameter, 2 in the center, that's what they call it, 9 plus 2, and the way probably was not discussed, even probably, I don't know, it was not discussed in Bio 1 that how that 9 plus 2 move, it is the same concept as you learned here during the sliding filament, right? Attack, pull, release, attack, pull, release. Same thing. Those 2 microtubules in the center, they attack, pull, release to the 9 parameter, and that's how the cilia move, wiggles back and forth. But we're not going to go into details. But that's pretty much the scientists believe. Pseudopodia, they have ectoplasm that transports or hyaline colloid outside of the cell, and then endoplasm, more granular, more fluid inside of the cell. Cilia flagella, 9 plus 2, you already know this is a transmission electron picture of it, 9 microtubules in the parameter, 2 in the center, and this is cell membrane. Okay, not all Protista have cilia or flagella. Same structure, both for cilia and flagella. Quick question from previous material. Did brush border have cilia or flagella? Brush border in your gut.
Cilia.
Did they have the brush? Remember microvilli? Microvilli, did they have 9 plus 2? No, they didn't. The microvilli in your gut did not have 9 plus 2. How about in your trachea, the cilia in your trachea, the ciliated pseudostratified columnar epithelial tissue, did they have 9 plus 2? Yeah, there was cilia right here. Fallopian tube of female. Yeah, you saw them on the microscope. It was all cilia. And they had, the only place in human that we have flagella is in sperm. There is no other flagella in human body. I'm talking about human body. Okay. Okay, Protista. Let's go back. Here are a few ectoplasm. It's more clear, transparent. Right here, this is endoplasm. Right here, and this is the nucleus, mitochondria, so on and so forth. The model, the other model was too heavy. I didn't bring it. I'm sorry about that, but that's okay. You guys are tough. You can look at the other model and learn it. Do you guys see that? Of course, this is cell membrane, the gray area. You see that? And then underneath there is a creamish area. Do you guys see that? That creamish area is ectoplasm. And then you have the gray area right here. And that is your endoplasm. And all of this middle area is your cytoplasm. Of course, this would be path for nucleus, this would be path for vacuole, this would be lysosome, you know, a few other structures. This is a pseudopod, you all know what I'm talking about. Pseudopod, and then we will talk about this in a little bit. So that's pretty much what am I referring to. I will talk about these other structures, contractile vacuole, vesicles, food vacuoles, excretion and osmoregulation. That's the next one. Okay, what happens, you guys, quickly, contractile vacuole. So in this model, we have one, we have another model in the lab. You know, I didn't bring it here. So this would be contractile vacuole. It was a lab practical exam question. Let me take the tape off. Okay, this is a contractile vacuole. And what is a small vesicles? So if you look at, you see these little gray dots in here. Those are vesicles. What happens with these organisms? Since the water outside is hypotonic, biology 1 stuff, and inside of the cell is hypertonic, based on Dr. Osmosis. He said this is a cell membrane, so water goes from high concentration, water goes from high concentration to low concentration. So if they do not come up with a mechanism to get rid of excess water, they will what? Pop. Explode. Pop. Thank you. They will explode. They will die. So what happens when the water going in by osmosis? These vesicles take the water. These vesicles they move and bring the water and dump it in contractile vacuole. Contractile vacuole becomes larger and larger and larger, and then contractile vacuole move to the cell membrane and dump the water out. Do you guys see that hole in here? That's contractile vacuole. So it attaches, in Bio 1 you learned it, that this is made of a phospholipid, right? Outside of it is phospholipid. Inside of it is full of water. That's what the function of vacuole is. It stores water or waste material, sometimes food. So what happens, this contractile vacuole moves and then attaches to the cell membrane and dump the water out. And this keeps going and going and going until the cell dies by no lifespan. Everything has a lifespan. You will see starfish is an exception. They live for hundreds of years. Okay, so that's the case of the top one. The bottom one is ciliated organism. This is a paramecium, example of a ciliated organism, the bottom one. So still same story, water molecules, concentration of water molecules is greater outside than inside of the cell. So water molecules constantly goes inside of the cell. I hope I'm making some sense. So if they do not have a mechanism to get rid of the water, they will explode. So the water goes through, I'll show you a picture, it's not in this model. They go through these rough endoplasmic reticulum. You know that from Bio 1. Inside of rough endoplasmic reticulum is a tube, we call it lumen, right? Lumen of rough endoplasmic reticulum. So the water ends up into these contractile, these ampulla. This is an ampulla. In this case, you have 5 of them right here. And ampulla dump the water into contractile vacuole. Contractile vacuole attaches one of the ampulla attaches to the outside, and then the contractile vacuole squeezes itself and the water goes out. I hope I'm making, I'll show you to you in a diagram in a minute. So these are ampulla. This is underneath the big ball, the orange ball, is contractile vacuole. And then right here, you see that little hole, that hole is called excretory pore. It gets rid of excess water. And this is more advanced. If we talk about evolution again, this is more advanced than the top one, okay? So this type of excretory system is more advanced than the top one, the one that I just mentioned. Right here, that's what I was talking about, you guys. I'm beating the dead horse again. So these are all our structures. Here is the endoplasmic reticulum. They bring the water comes from outside of the cell to the inside of the cell through the cell membrane. And then the endoplasmic reticulum brings the water through the ampulla, the red or ampulla, and dump the water into contractile vacuole. And one of the contractile vacuole is called excretory pore. One of the ampulla is called excretory pore, and then dump the water out. Okay, that is in case of Cilium. This is more advanced, and so on and so forth. What else in here I should mention? Well, you have the food vacuoles. Here is the gullet or oral groove, which food material goes through here, and they form a food vacuoles. And then the food vacuole dump the waste material, diffuses cell membrane, diffuses the cell membrane of the cilia, and then dump the waste material through cytoproct, the openings of cytoproct out. Trichocytes, the base of the cilia. And then cilium, as you know, is singular, cilia is plural, flagellum is singular, flagella is plural. Yes?
So the ampulla is connected to the reticulum and the membrane at once?
Right. Well, different ones. This is a different one than that one. If you go to YouTube, you might, that's where I saw it, you will see that these guys have two, Paramecium have two contractile vacuole. And it's just like, I don't know, like our heart. One pumps up, squeeze the water in, and then one, it goes like this. Am I making some sense, making an analogy? So this one is pumping the water out, this one becomes full of water. And then this one pumps the water out, and this one becomes full of water. Just like that. There are some nice YouTube video, you can watch it. And they usually have, in case of Paramecium, they have two contractile vacuole. Did I ask you a question? Or
Yeah.
are you in a tangent? Okay, nutrition, autotrophs, we talked about that, heterotrophs. Phagocytosis, it means cell eating. When a cell eats something else, you've seen that here in where? Where have you seen phagocytosis here so far? In this class. The what? Macrophages, but I gave it a name. You remember? Not my blind. Huh? No, they do not eat. Antigen-presenting macrophage, they do not eat anything. They make interleukin. But at the end, the policeman who comes, it was called
Phagocytic macrophage?
phagocytic macrophages. Yes, macrophages, but phagocytic. That's the term I forgot. Phagocytic. Okay, lysosome. You know from Bio 1, lysosome is full of enzymes, and they release their enzymes to kill the bacteria or whatever food material inside of the cell. And then there's a disease, rheumatic fever. I don't know if you've heard about that or not. You know, the lysosomes go out of wacko, and the person's hands, you've seen the pictures, in severe cases become deformed. So lysosomes are good, but the nickname for lysosome from Bio 1 is called what? Very good, suicide bag of the cell. Suicide bag. They call it suicide bag of the cell. Food vacuoles, of course, exocytosis, the waste material goes out. Cilia have cytoproct for expulsion of waste. Pinocytosis is what based Bio 1 again? Cell drinking. It's totally different than cell eating. Two different concepts. Bio 1 material. I'm not going to go over it. But, reproduction, fission is cell multiplication process when more individuals are produced. When one cell breaks down and becomes two cells, just like in mitosis, it's called fission. Not fusion, fission. That's the right pronunciation. Fission is when the cells are multiplying. And then binary fission, two identical individuals are produced, and budding is progeny is smaller than the adult. You might see it if you look at slides of paramecium in the lab. You might see a big, huge paramecium right here with cilia all around it, and then right next to it, you might see a little one, and you say, Amir, what is this? This one is budding. So this one, bud from the mother paramecium version, and this one eventually becomes as big as the original paramecium. So it's called budding. And you see it, I don't know, you saw it, I do it in my human biology class. We do it with yeast. You look at the see yeast on the microscope, the cooking yeast, the baking yeast that you do, and then you see budding in there. Multiple fission is possible, yes, it's true. Sporogeny, development of spores or sporozoites within oocyst. I might talk about it now or later. Schizogeny is when cytokinesis is preceded by the several nuclear division. So might as well, let's talk about it now, and then so we don't have to worry about it later when it comes to malaria. And this comes into malaria and epicomplexa. So let's talk about that right now. So while your brain is fresh. So what happens, you guys, in Protista when we have a microgamete, we call it a microgamete. I should put it in black. You have a microgamete, and then you have a macrogamete. Microgamete would be like a male, and then a macrogamete would be a female. When they come together, fertilization occur, fertilization occur. Then you have one cell, which this cell, they don't call it zygote, they call it oocyst. Do I make sense, everybody? And then, of course, if this one enters that one, how many nucleus do you have? One, right? In kindergarten, you learn 1 + 1 is equal to 2. But now in biology, you're learning 1 + 1 is equal to 1, right? Sperm plus egg, right here, I talked about it last time. Sperm plus the egg gives you what? One cell. Amir, I thought 1 + 1 is 2. No, that's for kindergarten. You're in college, you're in biology now. 1 + 1 is 1. Oh boy, tell that to 6 years old. Say, Daddy, I don't want to talk to you anymore. That's confusing. Okay, so that one cell, one nucleus, right? Then the nucleus inside of this oocyst, you will have a few cell multiplication. In this case, I put 8. Some cases there are 4 cells in the oocyst. You see what I'm talking about? Do I make sense? That process is called sporogeny. This, of course, is oocyst, sometimes called spore. This is different than the spore of plants. This is Protista spore. Okay, so what happens inside of that oocyst, inside of that spore, they call it sporogeny. Yes, ma'am?
So the nucleus just divides?
The nucleus undergo mitosis.
Okay.
Right. One nucleus becomes 2, those 2 becomes 4, that 4 becomes 8,
But
and
it's
stops.
Still encapsulated in that.
Very still encapsulated, and you will see that in here. Not maybe if not today, you will see it definitely on Tuesday. This is toward the weekend. I try to go easy right before weekend so you can enjoy your weekend. Okay, that's the case. Okay, then schizogeny is a little bit different when you have a host cell, nucleus of a host cell, and when one of these bad guys, have you heard that song? Bad boy, what you gonna do when they come after you? You know what I'm talking about? You've heard that song before. Any of you in here like that? No? No? None of you are here. I'm in a good place now. Shish kebab. I should buy lottery to get it.
I'm gonna take the fifth.
Oh, you're going to take the fifth on that one? Okay. So when the bad boys enter the host cell, am I making some sense? Then this one undergo nuclear division. 1 becomes 2. Then 2 becomes 4, right? You remember that? Final 1, 4 becomes 8. What am I missing? Mitosis. What is missing here? The what? Virus? No, this is the nucleus of one of these bad guys. One of these cells gain entry, one of them gain entry inside of the host cell, and then that nucleus, right, becomes 2 nucleus. 2 nucleus becomes 4 nucleus. 4 nucleus becomes 8 nucleus. What am I missing? Mitosis. Do you remember your mitosis? What is missing in here?
Cytokinesis.
Thank you. Cytokinesis is missing. Schizogeny. That's what schizogeny means. So first you have a whole bunch of cell division, and then later on cell membrane comes around them and cytoplasm is formed. That's what schizogeny means. So now you know the difference between sporogeny and schizogeny. You will see it, it will make sense. Types of gamete fertilization, syngamy, fertilization of one gamete by another, autogamy, gametes, nuclei formed by meiosis, fuse, and we'll talk about this, fuse to form a zygote inside a parent organism, conjugation, when 2 organisms come together and they exchange nuclear material, for example, in paramecium. Endocytosis and exocytosis, please read your textbook. I would like, this is a very important concept. I always ask essay question or multiple choice question regarding endocytosis. Let's go back, let's go one by one. I don't want to confuse you because it's important. What did I say? Unicellular form. What is it? The unicellular forms are amazingly successful in extremely harsh conditions due to various survival mechanisms. That's what happens in case of encystment and excystment. When the conditions of the environment is harsh, when a pond, in early days of Mother Earth, a pond, a river, nowadays too, with global warming, dries up. Okay, when there is not enough food in that pond, when it freezes, organisms undergo, they call it protozoa. Protozoa, I say organisms, I mean protozoa. Protozoa undergo encystment. It is equivalent to what you know about, almost, to sleep, birds sleep in the wintertime is called what? Hibernation. Here we go. See my mind. It's crazy mind, isn't it? Hibernation. They undergo hibernation. What happens, these guys, they don't call it hibernation, they call it encystment. So they are in the environment and their metabolism is slow. They do not do much of metabolism. They are do not divide anymore. They're just waiting for next rain to come. They're waiting for next food to arrive to the pond, lake, wherever they are, and then they can grow back. And that's what I mean. These guys are hardy. They form a cyst. We call it a cyst. Encystment, based on your textbook, is related to the ability to form cysts. This is a little bit different than what you're going to learn later on, a cyst that is in the brain of a human caused by Toxoplasma gondii. I'm not talking about a cancerous cyst. I'm talking about protozoa cyst. We call it bradyzoids later on. But anyhow, they form a cyst which are dormant. That's the key word. They're dormant. I said everything. I didn't say the word dormant. Dormant forms that shut down metabolism and have a resistant external covering. I didn't say that either. And then secreted by Golgi apparatus and not in paramecium. Does not have paramecium that do not undergo encystment. And you will see a few examples. In the lab, we have a model of euglena, a slide, a microscopic slide of euglena with a cyst. And then 2 by 2 slides, you have a few cysts. Okay? And then encystment, when they come out of this encystment, is called excystment. Okay, they exiting. I hope I'm making some sense. Encyst, they form a cyst, and exist when they get out of their encystment. Escape from the cyst when environmental conditions are favorable. Aubrey, am I making some sense? Important for parasitic forms. To survive the transfer from one host to the host to another host. You will see that in a few minutes. The phyla, these are the phyla, you guys, you should know this. I will ask on the microscopic slides, I will ask, we have all of these we have in the lab, except, anyhow, some of them. Well, we have the 2 by 2s of them. We don't have Dientamoeba. We don't have Chlamydomonas, we have wall logs. But anyhow, most of these slides, we have it in the lab. We don't have Eimeria, we do not have Babesia. But anyhow, you will see. You should look at the slides in the lab and you will see. And then you should know the name of the file. Okay, King Paul came over for good sex. You remember that? No? You don't remember that? I didn't say that? Did I say that? I didn't say that?
Will said it.
Oh, Will said that? You know, kingdom, you remember? Phylum,
King Phylum.
class, order, genus, species. Easy way to memorize it. King, Paul, K for king, kingdom, Paul for phylum, came for class. You all know what I'm talking about. King Paul came over for good sex. Good, genus, and sex and species. At the beginning of semester, I said in order to remember things, you have to have sex or blood and glory. You remember that? So here's the first time, the only time. The first, only, last. Okay, so you remember, we are throughout the semester, after this exam, then the next exam, you have to know the name of the class, not only the name of the phylum, the name of the class. For this exam, no class you have to know. Okay, even though it comes up in PowerPoint. I will not ask for any class, but the exam